Can everybody see the slides okay?
Looks good.
Very good.
All right. So again, thank you for having us today. So today I just want to share a little bit about our mission at the Phinizy Center and our work. I'll cover some of our Savannah River research and why it is important. And Jacob will tell you about our Rocky Shoals Spider Lily restoration project. The Phinizy Center for Water Sciences is a nonprofit organization that was established in 1996. As such, our mission is to provide leadership for sustainable watersheds and economic vitality through research, education, and by connecting people with nature. In order to fulfill that mission, we have three departments, the research department, which is myself and Jacob, our education department, and our park and administrative department. I'm sure that many of you know, have probably been out to the park. We have a beautiful nature park that our organization keeps open dawn to dusk year round. The park itself is 1,100 acres and has been referred to as a little Okefenokee. And we're recognized as an important bird area with many birders coming from all around to just see all of the different types of birds that you can see out in the park. And we're also home to some special constructed wetlands that are used for wastewater purification. In addition to a beautiful park, we have an active education department serving toddlers to adults with over 100,000 students coming through our programs since 1996. Our education department focuses on learning about nature, stormwater, and watersheds with the kids and allows children that may not have opportunities normally to have hands on experience outdoors to get those opportunities. And our adult learning classes include things like master naturalist, ornithology, and dendrology. So there's a lot that anybody can come and do out at the park, both at the park itself and in education. So, a little bit about the research department. In our research department, our mission is to study freshwater ecosystems in the southeastern United States with an emphasis on the Savannah River Basin. Through our research, we aim to find the necessary balance between a healthy river ecology and the needs of the communities. This is important because the needs of the communities are always growing, but we want to make sure that we have a healthy river too. To fulfill that mission, we've been studying the Savannah River Basin for 18 years now, and as such, we partner with local organizations to monitor water quality and provide data for management decisions. So, as part of that, since 2005, Phinizy Center scientists have studied water quality on a 140-mile stretch of the Savannah River from North Augusta, which you can see on this map here, from North Augusta all the way down to Clio, Georgia. This Savannah River Monitoring Project is a partnership with municipalities like Columbia County and Augusta Utilities. And we always like to try to work with other municipalities besides them as well. We have the most extensive and longest running data set on the Savannah River currently, which allows us to have a really good baseline for water quality on the river itself. So as part of this, we have continuous monitoring equipment at 5 sites, and you can see the sites in this right here are the little blue dots. Those 5 sites have continuous monitoring equipment that measure physical parameters, including pH, dissolved oxygen, temperature, and conductivity. And these monitor and take data every 15 minutes. In addition to those, we also take water samples for nutrient analysis each month from 7 sites, and we test those for things like phosphorus, ammonia, nitrates, nitrites, and organic carbon. So this is a little bit better of a map of our sites. These red lines here represent dams. The little blue dots are our sites. And then we also have stars for the Augusta wastewater treatment plant and for the Savannah City drinking water treatment plant. So, as I said, this map shows all 7 of our sites from north to south down the Savannah River. And each site was chosen to represent different land uses that affect water quality, such as agriculture and industrial inputs. And we also wanted to illustrate the different uses of the river itself and its importance in energy production, recreation, and for wildlife. And as I said, you can see here's the Augusta wastewater treatment, and you see that down here, the city of Savannah is taking in water for drinking. And that means that we need to make sure that our water is clean as it gets down to Savannah because they're using it for their drinking water. So if we wanted to take a look at some of our data, this box plot here shows dissolved oxygen concentrations at all of our sites for the 2022 water year. Dissolved oxygen is the available oxygen in water for biological processes. This is necessary for aquatic wildlife and plants, and many species require specific levels of dissolved oxygen in order to survive. So if we look at this and we compare the midlines of each box plot, we can see the dissolved oxygen is higher at our site near Hammond's Ferry, which makes sense because this is going to be near the Augusta Shoals, and we're going to have a lot of mixing of water going across those shoals, which is going to add oxygen into the water. And then we can see that it goes down a little bit as you go down the river, but you can see that we're steadily around a 9. The lowest values we get are around 5, and that's pretty good levels for dissolved oxygen. At those levels, we can have things like bass and other fish surviving, and they're not in any danger. This is just one example of how we can use the data. This is only one parameter that we look at when we look at a lot. And we can also make determinations of trends over the whole length of the project, which would be 18 years at this point, or we can just compare year to year, site to site. The possibilities are really endless with this amount of data. And to kind of illustrate that, this graph here is 16 years of water temperature data, and you can see the trends and the fluctuations with the seasons, and you can just get a really good picture of what the water temperature is doing over a whole 16-year time span. So why do we study the Savannah River? The Savannah River is important to everyone who's living around it. It provides drinking water to over 1 million people living in Augusta, Savannah, and on the coast. We use the river for recreation and ecotourism, as well as sustenance. And industries use millions of gallons of water every day. In addition to all of that, the river is home to many endangered species, such as sturgeon and the Rocky Shoals Spider Lily, which Jacob will now tell you a little bit more about.
Yes. So, can everybody hear me well?
Yes.
Good. Okay. Awesome. So, one thing before I wanted to start in on the Shoals Spider Lily, I want to give credit to where credit's due. There's another person that works with us in the research department. He's only part-time, works 1 day a week. His name's Owen. If I mention him later on in this presentation, he's the one that is heading up this project with me. He's a student at AU, so he had a lab tonight, so he couldn't make it. But I did want to, give him credit. He's a huge part of this project. So I just wanted to start out with really what is the Shoals Spider Lily. So the Shoals Spider Lily is just a really awesome plant, especially in this area. It was discovered by William Bartram in 1773 in Augusta, Georgia, on the Savannah River. This is the first place that he saw it. So it's just a really cool thing to be able to work on a project where this is the place where it was discovered and cataloged. So this lily, it is endemic to fall-line streams in Georgia, South Carolina, and Alabama, and only in those areas. It is an aquatic plant. There are other types of spider lily that grow in just regular soil, but this one in particular is the Rocky Shoals Spider Lily. It is only found in shoal areas. So this flower is also, or this plant is also threatened in the state of Georgia. So it has that designation. The populations have been on decline for years now, mainly due to the building of dams and deer extirpation. So deer really, and Rachel, could you go back real quick? Sorry. Deer really like these plants for whatever reason. They will swim out into the river to get these things. Um, so there's a multitude of factors, and I'll go into later on in the presentation on like how dams affect them. And I also did want to mention the peak bloom season is about mid-May to late June. They're really beautiful if you've never seen them in person. A really great spot to see them is at the head gates of the canal, right below the diversion dam spillway. There's a population that you can see a picture of. Part of it, the center picture on this slide, is that population right below the spillway, and it's really beautiful. The white blooms really pop. Just wanted to let y'all know that. All right. So, why are they important? They are an indicator species for stream health. So their presence or absence, just at a quick glance, you can tell how, if that stream system is healthy or not, at least in these shoals area. So we know that they will not be in anywhere that is even remotely polluted. So it's great that we see them on the Savannah River, in Stevens Creek, Broad River. It's just really great that we're seeing them there because we're treating our water correctly around here. So it's just really great to see that. And then they improve the water quality as well. So like any other plant, they take carbon dioxide in, push oxygen out. So when they do that, they push that oxygen into the water, increasing the dissolved oxygen content for things like aquatic insects and fish to use. And it's just really good for the river system. So they also trap sediment. So it helps with sediment and nutrient transport. Nutrients will attach to the sediments since they're both charged particles. So it helps with nutrients depositing and not and like decreasing erosion down the river by having that sediment get trapped by the plant and kind of stored where it is. And then the plant in turn also uses those nutrients that are in the sediment to grow. So, and one of these nutrients is phosphorus. So they lower phosphorus in the water. They actually uptake most of the phosphorus that they're going to use during the blooming season. When they're blooming to create those blooms, their uptake in phosphorus increases a lot during that period. And this is good because while it's not the only factor that contributes to this, if you have too high of phosphorus in the river, it can be a cause of algal blooms. Again, it's not the only cause, but there's an increased chance when you have very high levels of phosphorus. So they also provide good habitat structure. When we're out on the river, when we've been doing this project, we've seen tons of moths, macroinvertebrates like the stonefly that you see in the bottom right-hand corner, tons of dragonflies, mayflies, whatever you want to see, water snakes, birds. I've seen ducks in them when we've gone up. They're just an incredible habitat for just a multitude of things. The fish love it. So it's just a really important and valuable asset to the river in that habitat. So, and then lastly, it encourages ecotourism. These Rocky Shoals Spider Lilies are only found in Georgia, South Carolina, and Alabama, and only in these fall line streams. And they were discovered here in Augusta. So it's one of the few places that you can see really actually pretty good populations. While they have been declining, some places are definitely worse off than us, but it's a good draw in for people to come here, experience them, like see them up close. So it's all just great for it. So, for this project, we had known about the shoals lily in the river as the research department. Um, kind of knew that the populations were declining. Had um, seen some papers from some people that um, from a professor at AU that she had done about 10 years ago. Um, and so we were just thinking like, man, this is a threatened species. We should do a project on this and try to really restore these populations that are in such decline. So we did. We started researching shoals lily restoration, and we found a master's thesis from a Clemson student, Andrew Grunwald, who had done his study in Stevens Creek in South Carolina on the best methods to restore shoals lilies and rear them to where you can have viable populations, restored out on, natural creeks and rivers. So we saw that, we loved it, and we were like, let's try to recreate this. So we wrote a grant to the Porter Fleming Foundation, and they funded us for it. So we started this restoration project, and it's just it's been a blessing to work on during this time. So for our project, we took his master's thesis, went through all the different kind of facets of what you need to look for to make this plant thrive. So we took basically all of his best work and funneled it into the best way to restore these populations. So in that, we did habitat assessments, water monitoring, restoration, and like what I'm doing right now, what we are all doing right now, community outreach. It's arguably the most important part of this is having y'all and the public just aware and willing to help and listen and learn. But I'm going to go through each one of these in the coming slides. So for the habitat assessments, we had 5 shoals that we were monitoring on the Savannah River. The first shoal site is right below the spillway at the Augusta Canal. The second shoal site was probably about 3/4 of a mile down. It's the start of an island. It's above I-20, but below the spillway. And that population, from the paper that we read from Donna Weir at Augusta University, when she did this like 10 years ago, there was apparently a very large population there. Well, when Owen and I got out there were literally 2 plants left. And we were like, all right, we've got to make this our restoration shoal that we're going to restore and try to build back this population. So that was our restoration shoal that we wanted to build back up. And then the third shoal, if you're going over the I-20 bridge into South Carolina, you can actually look out the right-hand side, and it's across the clearing on the canal trail. You can see it from both of those spots, but there's a large population of spider lilies right there as well. And that's our third site. And then our fourth and fifth sites had no shoals lilies at all. We only found 3 sites that even had them. So there were 2 shoals that we picked out that we were going to use that we were going to take all these parameters that we have listed here and see which one matches where they're actually growing the best to start a new population of shoals lilies there. We ended up picking our 4th site. It's about a half mile, maybe 3/4 of a mile down from the 3rd site where the clearing is. And then the 5th site was near the pumping station, like in Eisenhower Park area on the river. But we did not end up using that 5th shoal site. It didn't end up really having all the parameters that we wanted. So when we were out there, we basically took a 40 meter by 50 meter square of the shoal and every 2 meters had a half meter by half meter block, and placed it down and went through these parameters in each one of those. So at the end of it, we had 100 measurements. So we would do percent coverage of spider lilies. So if we put that box down, how much of this box has shoals lilies in it? And the goal with that was, once you're done with your transect lines and sampling, you can use the percent cover to do a population estimate in that area. And then in that same box, we would do substrate class. So the substrates are on the screen. At the top is fine sediment, then coarse sediment, and then bedrock. What we did not want to see wherever we were was really the fine sediment, or at least that being the major sediment there. Because when the shoals lily take root, they need something that won't be swept away in high flows, since this is fast flowing water. And if it's sand, they can't root in and they'll just blow down the river. And then, so we were really wanting coarse substrate and bedrock. The bedrock was a little bit trickier. It had to have like cracks in it that it could wedge itself into. But from the thesis, the best substrate was the coarse. Maybe not as fine as it is pictured on the screen, but if you see like the granite rocks on the side of like streams or anything like that, that's about the size that you're kind of shooting for. Big enough to not be knocked around by the river during high flows, but small enough to where it can be wedged in easily and the roots can like anchor to it. So we were looking at substrate class, and we classified it, based on fine, coarse, or bedrock. And then in each one, we did water depth and flow. So I said earlier about how the dams are so like important in this process. So from Andrew Grunwald's thesis, he found that the shoals lily's ideal depth of water was in between 5 and 20 centimeters. And that is, barely anything. That is such a small range for the ideal. They can definitely do a little bit less, do more, but the ideal range for very healthy populations was that 5 to 20 centimeters. And we had very similar results to him when we did our work out here, in our habitat assessments. So we would take water depth in the area, and then we have a flow meter to where we could see the velocity of the water coming through. And we used all of these parameters to decide just how the habitat is in each given area, where there are shoals lilies. But then these were also the determining factors in deciding where the new population was going to go in those two shoals. So the next stage in this project was water monitoring. So we took depth measurements when we went out there, sampling, for these habitats, but we wanted long-term data. We wanted the day in, day out. So we installed 5 depth monitors out on the river. They take readings every 15 minutes. So whenever I go out and collect this data, we can bring it back and we can see exactly what the level is staying at in these shoals at base flow conditions. And that's really important information for the longevity of, I think, the species and future restoration projects. It gives you a really good insight of exactly where and what water level they're kind of staying at. And then also in this, just like the Savannah River project, we're doing water nutrient analysis. So we're doing nitrates and nitrites, which these plants will uptake. We're doing organic carbon, which is leaf litter or just organic matter in the river. Phosphorus, which is, as I mentioned earlier, very important for the shoals lily, and then ammonia. And we run these quarterly. The depth monitoring is all the time. nutrient analysis is quarterly out on the river. So the restoration part, you can see a picture of Owen here. He's the person that's been mainly out there with me and in charge of this project. So for our restoration, we collected 1,600 or a little bit over 1,600 seeds out there. That did not put a dent in the amount of seeds that these things make. Um, as you can see in the bottom left-hand corner, that's what the bloom ends up turning into. So where the bloom is, the seeds come out and they form these like pouches, and then they weigh the plant down so much that it droops over. These ones weren't drooping into the water because there was a rock in the way, but they're supposed to droop into the water, and then the current, once they're ready to break off, will break them off and then hopefully wedge them into a rock to where you can get a new plant started. So we collected them when they were in this stage. They're about the size of a golf ball or so. They're actually pretty large. And the best way that the thesis laid out to rear them to, he tried planting them just as seeds, growing them into seedlings, and then growing them for 1 year into a full plant and then planting them. He found that growing them into seedlings had the best results. So that's what we used for our project. The second picture where that blue pool is in the background is what a seedling looks like. So there's roots here at the bottom coming off of it. And the seed is attached where it's being held over here. But the roots are attached to it. There's a bulb, and then there's some leaflets coming up. So that's what the seedling looks like. Once it's larger than that, he found that the roots didn't want to anchor and they would be swept away. Whereas in the seedling stage, the roots would go into the rocks and anchor it there and keep it there. So when we went out to plant them, the picture of Owen there in the middle is the last two seeds that we ended up planting out there. We ended up planting right at about 1,400 seeds. About 200 didn't make it due to mold or disease or something, and we had to take them out. But we ended up planting 1,400 between the restoration shoal and the new shoal site. So he also found that the wedge method was best. So he tried several different methodologies in his paper where he just broadcast them out into the stream like they would be natural, where he wedged them like what we ended up doing, and by kind of interlacing them in a burlap sack and tying that to a rock so that they have an anchor point. But the problem with that one was that he found during high flows, the river would rip that off and take them down. So it turned out the most natural method of how they would reproduce, which is the wedge, was the most productive for them. So in the bottom right-hand corner, we have a photo that was taken. We planted these probably, I think it was about 3 weeks before this picture was taken. We had wedged them into some bedrock here, and it had started out when we first planted it 3 weeks prior as one of those little seedlings with probably about a 3-inch leaf or so. And when we came back, it had anchored into the bedrock, stood up straight, and had about 11-inch leaves on it. So when they anchor, we could always tell whenever we had seeds anchor when we went out, week after week, because whenever they did, they would shoot up. And it's just been really awesome to see. We won't know for sure how many of our seeds made it, that we planted until next spring. They have to get through this winter cycle for us to get the accurate number of how many exactly survived it. But from what we've seen last time that we went out, I think that we have probably about 200 to 300 seeds at our restoration site and the newly established site in each of those that had taken and at least grown some since we had planted it. Um, we're hoping for a pretty good survival rate over the winter, but we just gotta wait and see. So the last part of this project is community outreach. We love doing community outreach. We love telling people about this project. Um, we had some social media posts when we were doing all the planting and seed rearing for the project. Um, really, what it boils down to is we need to take some more pictures and get out there and show you how they're doing now. But one thing that we also did, the pictures on this slide are from a seedling planting demonstration that Owen and I did. Um, we took a group of interested individuals in this project up to the first site that we're at. So right below the spillway at the canal, and got to talk to them about the project, got to bring them right up next to the lilies. They could see them up close. A few of them still had like the seeds hanging over on them. And we got to tell them all about the project. And then we brought 50 seeds up there um, and let them plant them, in the shoal and really get a feel for, the work that we're doing and just to really get a hands-on involved approach to it. Um, so I mentioned that to say for our future events, we will be doing in the spring of this coming year, uh, more kayak trips out there onto the river. If you or anyone that are interested, we'll be going out there and looking at all of our sites that we've done, seeing how the established sites are, and then seeing how the new sites that we're trying to establish are doing as well. So we really just want the community to be out there with us and experience it and really be up close to them. They're just so beautiful when you're just right there. Um, it really does make an impact. And then also in the spring of 2024, it also, it says spring of 2024. It may be more leaning towards like a summertime because we want them to be in bloom, which is mid-May to late June. So somewhere in there is when we're planning for these. Um, we'll also have a research symposium. We'll have more information on that at a later date, but we will have basically the final data on this project. We'll go back out, see how our seeds did at the site, and then have all the water level data. And really, it'll be a presentation on all of our data, all that we've learned, and really to get the community involved, educated, and aware. And I mean, that's all that I can ask for. I mean, thank you to, y'all that are here right now, taking the time to like listen to this and, really like supporting us for this project. So I'm going to turn it back over to Rachael to tell you about a few other. We have a bunch of research projects. She's going to tell you about a few more of them. Ways to get involved at Phinizy, and then we'll be more than open to questions. So
yeah, like Jacob said, we have a lot of other interesting projects going on at Phinizy. We also have our Eco Explorers experience, which is a partnership with Destination Augusta. And in that experience, you get to put on waders and sample for macro invertebrates, and it's a really cool experience that you can sign up for. We also have done work with South Carolina DNR monitoring sturgeon movement in the middle Savannah River. That's another species that is threatened and endangered in the Savannah River. We've done projects recently examining the sources of fecal bacteria in Brushy Creek. That's the hog picture here. It was a project trying to determine if hogs were contributing to the bacteria in the creek. And we also do urban stream assessments in the Augusta area in a partnership with Augusta Utilities. So if you see our truck around town, we're probably out there in waders somewhere taking some water samples. And there are lots of ways that you can get involved with Phinizy. Like I mentioned earlier, we have a lot of educational opportunities from adult learning, or if you know people with kids, grandkids, we have lots of children's opportunities. We have events at the park, and we're always looking for park volunteers as well because the park is really maintained by volunteers. All of our boardwalks and things like that, we have to have volunteer help with all of that. And you can get involved in research, like coming out to our events that we're going to be planning for the spring of 2024, just supporting research and bringing us to put meetings like this where we can present our research and talk about what we do. That's really important, like Jacob mentioned, to have that community outreach and get people to know what we do here at Phinizy. But we will be happy. I'll stop sharing my screen now. I'll turn on my camera so we can answer questions. Oh, okay.
Great presentation. Really enjoyed it.
Thank you.
Thank you.
Very good. Very good.
Sturgeons, are they any locally able to reproduce? I mean, how far up the river can they go these days?
Yeah, so they do have reproducing populations here in the Savannah River, both of Atlantic and short-nosed sturgeon.
Hmm.
They go up right to the base of the New Savannah Bluff Lock and Dam. They can't get past that point. Their historic breeding grounds were the Shoals area, but I've gone out with South Carolina DNR on their boat to try to catch some of the sturgeon to tag them, and there is a sandbar right behind that dam that meets the requirements for their eggs and breeding, so they still have them breeding here.
Great. What percentage of your lilies are consumed by deer?
Oh,
Does that vary from spot to spot?
yes. Yeah, it definitely varies. So the the one nice thing about the two bigger populations that we saw is the one under the spillway at the canal, and then the one that you can see by the I-20 bridge. Those two are in the center of the river and don't have many access points for deer like around them.
Oh,
So we haven't really seen much extirpation at those sites. But that second site has a large island right at the tip of it, um, that deer live on, and tons of easy land access. And we only saw two plants out there the first time that we went out there. The next week when we went out, they were eaten down to stubs. We were just like, "Oh, come on." But what is nice, I don't know why they didn't completely kill the plant, but last time, my partner Owen and I were out there, um, for this, partner for this project, we're out there. Those plants that had been eaten had actually grown their leaves back at least somewhat.
oh,
Uh,
yeah.
so that
Well,
that,
that
that
happens
was
in my yard too.
yeah.
Any,
But that was encouraging to see that they can, grow back from it, but usually they eat them down to where they can't even do it.
any
So yeah,
chance of building a big fence?
I'm sorry.
Any chance of building a big fence?
So that's what we're working on right now over the winter months,
Ah,
is working on not building a fence, but we've asked around at different ways to manage these deer from like getting out there. And the best solution that we have seen is this, I mean, it's kind of fencing, but you stick these posts in the ground, and there's a type of like ribbon kind of rope that you soak in a deer repellent, and so it's like minimal out there. and you can put it along the edges of the property, and the deer will not cross it. It's what's used for people on hunting properties to create food plots on the land.
Hmm.
And for those food plots to grow and establish, you can't have the deer eat them right when you plant them. So they use this, fixture out there to keep them away. And we're looking into getting permissions from local landowners. And we'd have to get permission from the state to put anything
Yeah.
on the islands out there, and permission from private to do the other stuff.
Thanks.
Yeah, of course.
More questions?
I have one. The kayak trip out to see the lilies.
Yes.
Is that going to be listed like on your website, or how are we going to know when that happens?
Well, if you want to send me your email, I can make sure that you see it, but I think what we're going to end up doing is making Facebook and Instagram posts on it.
Okay.
I don't know necessarily if it'll be on the website, but we'll definitely make social media posts on it.
Okay. I'll send you my email after this.
Yeah, that'd be great. Do you want me to just tell you my email now, or find it on the Phinizy website, or?
I can get it.
Okay.
Or I can send it to Ruth, and Ruth can send it to you.
Oh, yeah.
Just how tall are those plants?
They get taller than you think. I mean, the portion that sticks out of the water is probably a good 2.5 feet at least, maybe even higher. I mean, there were some of them that were probably up to mid-chest on me at the tips of them, where the flowers were blooming. So I mean, probably 4.5 feet, maybe 5 feet.
Wow.
They were pretty tall. I mean, I'm 6'2", and they came up to essentially my chest. So those are the really large plants, though. Most of them are probably in the 3.5 foot range. And one other thing, their own seeds are not their only form of reproduction. They can also propagate. So they can send off offshoots, through their roots, and spring up another plant. The seeds is basically a backup for genetic diversity for that plant. So.
More questions?
Guess not.
Well, I want to thank y'all for this presentation. This was really great, and really learned quite a bit here. And,
Yes, thank you.
yeah.
Thank you for having us.
Yeah, thank you all so much, and thank you all for hearing us.
Yeah, that was really great. Thank you, Doug, for finding them.
Yeah.
Very good. Yeah. All right, what we're going to do now is we usually have a little business meeting after this. You're welcome to stay on for it, but
Yeah, right.
or you can or get out of here. But thank you very much. Really enjoyed it.
Yeah, thank you for having us.
Yeah, thank you.
Thank you.